임란, 저는 오늘까지 그 의사과학자 양성 사업이라고
그거
그러니까
30년 전부터 한 거 아닙니까? 의사과학자 양성사.
의사과학자 양성 사업이니까 의사 뭐 전공이나 뭐 의사들 신진 의사들 돈 주는 사업인데 그걸 학교에서 대표로 한 명이 써가지고 그 단체로 받아오거든요.
네, 원래
그
그렇죠.
대표로 쓰는 사람이 제가 쓰고 있습니다. 그러니까 저는 연구비를 일원도 못 받고요. 그 전공이라든지 뭐 이런 사람들 저 펠로우라든지 연구비 주는 사업 그래가지고 저희 학교 10명 거를 한꺼번에 받아오는 잡일이죠, 완벽하게.
그, 이번에 좀 슬픈 소식이 있어서,
왜요?
학회 지원금 지금 학회에서 보통 그 학회에서 주는 출장비 있지 않습니까? 그거,
학회에서 출장비를 줘요?
네네네, 저는 속한 학회에서 출장비를 주는데,
예,
이번에
임상학부니까
규정,
그렇지, 임상.
그러니까 이번에 규정이 좀 바뀌었습니다. 그, 면허 없는 사람은 출장 지원비가 안 된답니다. 학회 거기 정회원으로 섞고 있어도. 올해부터 그렇게 바뀌었습니다, 규정이. 그래가지고. 아, 그래가지고 지금 해외 트래블 펀드를 지원해야 되나 말아야 되나 지금 그렇게 고민하고 있어가지고.
그거 학회 출장이라면 학회 참석도 출장인가요?
네, 그 저기 포스터나 아니면은 오랄 발표에서
아, 그걸 출장비라고 그러는구나.
네, 출장비 지원이라고 하니까 한 800에서 800만 원 정도 지원하는 것 같거든요. 미국 가면. 근데 그게 작년까지 지원이 됐는데 올해부터 지원 안 된다고 지금 규정이 바뀌어가지고 그 면허가 있는 사람 면허 번호가 있어야지. 그러니까 그 의사, 간호사, 그다음에 물리치료사, 뭐 기타 등등등등 같은 의료 계열의 면허 번호가 있는 사람
아니, 회비를 내자는 회비.
내죠. 당연히 저 평생 회원인데.
회비를 내는데 하긴 일반 학회는 회비를 내도 출장비 이런 거는 전혀 절대 안 주지.
그래서
의의가 없지.
작년까지 지원이 다 됐는데 학회 발표하러 가면 지원이 됐는데 올해는 기초 쪽 그 면허 번호 없는 사람은 지원 안 된다고 그래가지고 지금
그런 게
올해
어딨어.
올해 그냥 그 트래블 그랜트 다시 지원을 해야 되나 지금 그거 안 하는 거
아,
예.
주면은 다 줘야지. 누구는 안 되고 누구 그런 게 어디 있어.
그래서
그거는 완전히 불공평하지.
뭐 이렇게 민원 넣을 수도 있는 것도
똑같이 회비 내는 사람들인데.
아니 근데 그게
그렇잖아요.
다
똑같.
예. 이게 보건복지 규정으로 바뀌었답니다, 지금. 그래가지고.
보건복지 규정에 그런 것까지.
네네네.
아니, 학회는 보건복지부하고 뭔 관계가 있냐. 뭐, 이해는 좀 안 되는데.
뭐, 그렇긴 한데, 작년까지 지원은 받는데, 뭐, 야, 제약회사 뭐, 어쩌고저쩌고 해서 뭐, 스폰서 받는 걸로 해가지고, 이렇게 된 것 같은데, 안 됐다 그래가지고, 좀 슬픈 사연이. 그래서 지금, 딴 트래블 그랜트 지금, 어플라이 시켜야 되니까, 저도.
음. 저희 팀 다 들어왔는데 시작할까요?
네,
오케이.
시작하세요. 지금 우리 쪽 학생들 지금, 아직 1월 때까지 가야 되는데, 지금, 안 하는 건 줄 알고 있어가지고. 아,
음.
이제. 알겠습니다.
Imran, can you start your presentation?
Yes. Sorry. my screen visible?
응,
Yes.
For some reasons, I cannot make it slideshow.
I'm sorry.
Okay.
음,
Good evening, everyone.
음,
I will be presenting our data, which was collected by uh, the single cell RNA sequencing analysis of the mouse liver tissue, which was treated with exosome. The exosomes were treated at 8 hours and 48 hours, and also with the PBS control. And then the liver tissue was collected and which was sent for single cell RNA sequencing. Uh, After the sequencing in the raw data, uh, we found almost 30,000 cells, which was part of the raw data. When we did the quality control of the cells, most of the cells uh, most of the cells contain the average genes around 2000 and the average RNA around 10,000. But there were some outliers where some cells contain around 6,000 genes and some cells contain around 40 to 50,000 RNAs. So in order to make a good data, we had to remove these outliers, and after QC we had around 25,000 cells, with each cell containing minimum of 2,000 genes and around 5,000 to 6,000 of RNA.
I have one
After
question.
Yes,
So you said the outlier, the minimum outlier represents higher the gene number of the higher RNA count, right?
yes.
So why the some cells showing the higher amount of the genes or RNA genes or the uh, RNA number?
uh, Mostly that is because of the precision of the machine. Sometimes two or three cells are
Ah, I see.
clustered with
I
each other,
understand.
so
Oh,
it is
okay, right.
counted as one. So
Okay.
we have to remove that one.
Because this RNA-seq machine operated by the, what, 10x, right?
Yeah,
10x,
10x genomics.
10x genomics. Oh, okay.
Yes.
So 10x, why in 10x genomics, the studies, uh, some analysis contain multiple, the number of cells in during the one analysis. Why?
Actually, the machine does not know which cell is which. In the sample preparation, every cell is given some specific adapter. So the machine only reads the adapter, and on behalf of that, it says uh, like uh, like hepatocytes adapter is called X. So every cell which shows that trait will be given into a cluster which is called hepatocytes, which we will confirm later. Do you understand?
No, I didn't
Okay.
understand. My
Every
question is why in 10x genomics analysis, why
Yeah.
some single cell seq shows the data from the multiple cells? You understand my questions?
Yes, I understand your question.
Because which make the outlier data contamination from the correct one, right?
Yes, yes.
So why? You don't know?
Actually, I to my best knowledge,
Uh,
every cell is given a barcode, and the
yes, you
machine
already explained.
yeah machine reads the barcode.
Yes.
So some cells are too close to each other, so the machine reads them as only one. I think that
Ah, so in the 10x genomics study, every cell marked by the barcode,
yes,
right?
yes.
But some sequencing read one barcode from two more than one cell, right?
Yes. Those cells are too close to each other. Machine
Two
thinks that,
cluster. That
yeah,
means with only one barcode, two
yes,
or more cells are making
are
aggregate
read as one, yeah. Because the,
1 year.
yeah,
I
it's
don't
on the
know.
machine precision, mostly 10 to 15% of the every read up in every kind of sample. We
Uh,
have to remove them because it is not completely precise and it can affect our downstream analysis.
okay, right. So multiple cells cannot be analyzed in single cell
Yeah,
sequencing. Okay.
yeah.
So you
After the,
remove, okay,
okay,
okay, and
I
then,
I remove the outliers, and then based on the barcodes, we we have to find the clusters, like all the cells which have same barcodes, they will be,
and other question. Also,
yeah.
you remove the dead cell or cell debris from your
Yes,
data.
yes.
How?
I'm sorry, I did not add it here. Any cell that has a mitochondrial percentage of more than 20% is
Mm.
is collected as the dead cell. So
Higher
so
the data with a higher mitochondrial DNA contents
yes,
means considered as dead cell, right?
yes, yes.
Okay.
So
Mm.
in this QC, I selected only those genes which have minimum of 2000 genes,
Mm.
minimum of 5000 RNA, and
Mm.
maximum 20% of mitochondrial genes, which is
Mm.
considered normal globally.
Okay.
Okay. After that, I did the cluster identification, and I found total 12 clusters. Uh, but at this time, we don't know which cluster is which. But after performing the differential gene expression, we can find the type of genes which are highly expressed in each cluster. So in this dot plot, I have shown 5 genes from each cluster. So these genes are highly expressed in separate clusters, and they don't overlap anywhere. That means they are specific to that cluster only.
Okay.
And
So,
now,
but how
yes,
did you make the cluster? What kind of method did you use for making the cell cluster? You
uh,
said the 11 clusters
yes, we
is
have to
identified.
perform the principal component analysis and unique molecular identification patterns, which is called the UMAP plot. So
Mm.
it will find only those cells which are unique than others. So it will cluster all the similar kind of cells into one place, and it will designate it as a cell type.
Okay, I didn't understand completely, but
It
before
is
making the cluster, cellular cluster,
yes,
you try the principal component analysis,
analysis.
right?
Yes, yes.
What is the principal component analysis?
In principal component analysis, we can find the cells which are located with each other. Like there is there is different clusters we will get.
I didn't understand. The next time, please explain how can you do the principal component analysis. Actually, when you are doing the PCA analysis, you introduce AI-based the AI-based algorithm, right?
Yes.
Still, I can understand how AI-based algorithm can make the principal component analysis. Next time, I
I'm
don't.
not. I also don't know much about it.
You should know because you are a student, right?
Yes. Yes.
Okay,
Oh,
okay.
Yes.
Lim
Yes.
if you want to know about how AI works, please give me your data. I'll explain how it works. If you need some help from my lab, as we have a AI analyzing system and AI analyzing protocol. So if you have a question, explain this. I'll gladly help you how to understand these things. So
Yes.
okay.
Mm,
Thank you. Yeah,
mm.
thank you. I appreciate your help.
Okay. Then next.
Okay. Then
So
based on
each cluster have a unique gene
cell
expression
type,
pattern,
yes,
especially five the highly expressing genes, right?
yes.
Okay, next.
Okay. From the from the available cell marker database, I
Mm,
found the universal marker of each liver cell type,
mm,
which I collected from these two databases. One is called Penglao database and one is called Cell Marker 2.0.
mm,
Based on these markers, then I tried to find the expression of each of these marker in each cluster.
mm,
So for the hepatocyte markers, I can see that the hepatocyte markers are highly expressed in cluster 0 and 4.
mm,
And for stellate cells, they are expressed in cluster 10. For
mm,
macrophages in 2. For
mm,
dendritic cells in cluster 9. And Kupffer cells, since they also work as the immune cells, so they have co-expression with macrophage and cluster 7, but
mm,
it is but since these markers are specific to Kupffer cells, so we call them Kupffer cells.
mm,
And some resident or recruited macrophages along with neutrophils and liver sinusoidal endothelial cells, B cells, T cells, and cholangiocytes.
mm,
Based on all these expressions, I confirmed the cell type markers and then redid the UMAP with
mm.
each cell type as here,
Okay.
and the dot plot of all the markers which is specific to each cell type.
Mm,
Since there is no overlap, that means the analysis was statistically correct.
mm,
And then and then I subset the data into two parts: one hepatocytes and non-parenchymal cells.
mm,
And in all these samples, the cell distribution, hepatocytes were in the greatest number as expected, and in the non-parenchymal cells, macrophages were the most majority of the cells. And if you see at the distribution of parenchymals and non-parenchymal cells,
mm.
uh, it is it is almost 50-50 in the 8-hour sample, but there is less distribution or not equal distribution in 48-hour sample. But
Yes.
the
So
cells are,
I have one question.
yes,
Actually, the live intact cells adequate for the single cell sick analysis. So
yes,
the number of hepatocytes is higher in the 8-hour sample
yes,
than the 48-hour
48
sample.
hours.
Why this happens?
It can happen because of the cell collection or cell transport, or they can die of
Right,
stress
right.
in the process.
48 sample, actually the 8-hour sample, the prepared at same day or similar times, right?
Yes,
And also 48-hour sample is also similar, uh, captured, select,
in
purified
same way.
similar, but actually in 48-hour sample, the storage of or transfer is at the same day we transfer the older sample. So maybe storage is some problem in 48 sample, right?
Yes.
Mm, okay. Anyway, so 48 sample, how many the hepatocyte can we analyze? How many?
Uh, in
You
hepatocytes,
know, maybe
around, I
around.
think it is between 1,000 to 2,000 cells.
Really? This is 1,000 to 2,000? Really? Oh,
I
okay,
think,
okay. Then it'll be good enough for
hopefully.
analysis. Mm. Actually, that means you know the storing or transfer of a purification of a hepatocyte, primary hepatocyte is not the easy.
Yes.
because the hepatocyte is easily damaged by from the variety of conditions.
Stress. Mm.
Easily, very, very fragile
Yes.
hepatocyte. Mm. Not easy. Okay, next.
Okay. Then I perform the differential gene expression on hepatocytes, and here is the representation of the differentially expressed genes at hepatocytes treated with exosomes at 8 hour,
Mm.
8 hour and 48 hour by both volcano plot and the heat plot.
Mm.
And the cutoff for fold change was used to 1, and p-value should be less than 0.05. So we can see that the significant genes, if exact number, we can see there are 430 differentially expressed genes at 8 hour and more than 1000 genes at 48-hour sample.
Oh, this number is sum of the increased and decreased genes?
Yeah,
Or,
they are both.
okay.
Okay.
Mm.
Based on the differential gene expression, I performed the gene ontology and gene set enrichment analysis of the hepatocytes. At the 8 hour sample, we can see that the most of the genes are involved in either in inflammatory response. It is acute or defense response or response to stress. We can also see that the enough population of the genes is enriched in the 8 hour sample in the enrichment plot.
Okay.
And the top enriched pathways were acute inflammatory response, defense response, and response to stress at 8 hour treatment.
Mm. So actually, the I know the gene ontology analysis.
Yes,
Actually, uh, uh, it calculates from the gene set
yes,
in each condition, showing decreased or increased the response, the expression, right?
yes.
But how we can we uh, so what is the difference between gene ontology analysis and enrichment gene enrichment analysis?
In gene ontology, the
Uh,
software will take the list of the genes and
mm,
it will find out the multiple pathways which the gene is involved in.
right. So
They can be
for the
common in,
geo analysis, we use gene set,
yes,
just only number
yes,
with showing the difference in state, only showing the statistical difference, right?
yes.
Okay. And for the gene enrichment analysis,
In gene set enrichment
gene
analysis,
set on enrichment
yeah,
analysis.
uh, the software will calculate the enrichment score by
Mm.
which in which pathway the gene is involved and to what extent the gene is involved on
Right.
beta
What extent?
yeah,
That is the important point.
yes,
For the major mixing the what extent of gene expression, we need to add the expression value, right?
yes,
Not only the no, name of genes.
yes,
So set enrichment assay, collective data show, including the name and their name of genes and their expression level, right?
yes, yes,
So in some terms, the gene set enrichment analysis is more accurate
yes,
for evaluating our data, right?
yes.
Okay. Okay. Good.
And
Next.
then gene ontology and GS EA for 48-hour sample. And
Mm.
most of the enriched pathways are related to translation or ribosomal RNA metabolic process.
Mm.
And the top two enriched process are one is related to cytoplasmic translation and another one is translation, which indicates that there is a difference between the effect of exosomes at 8 hour and 48 hour.
So that means 48 hour treatment after with the EV, the RNA, RNA, right? RNA,
Yes, mostly RNA
RNA transcription and protein translation genes
is highly,
associated with both pathways are increased?
yes,
Increased?
yes,
Okay, I want to see. I should know about that. So at 48 hours after the EV treatment, the hepatocyte increase the genes associated with transcription and translation, right?
yes.
Okay.
I
Mm.
have performed the analysis till hepatocytes only.
Mm.
I'm still working on the non-parenchymal
Mm.
cells
Mm.
since the type of cells and cell number are higher, so it takes a lot of time, more time than hepatocytes to analyze.
Okay.
So
But
today I'm
when
presenting
you
this.
analyzing non-parenchymal cells,
Yes,
you can also divide the parenchymal, non-parenchymal cell into, uh, you know, the liver resident cell population versus
immune cell.
immune cell. Immune cell is always introduced from the blood, right?
Yes,
So please divide. That means
liver
beta.
resident cells and immune cells.
Right. Kupfer cell, sinusoidal endothelial cell, stellate cell is resident cell in liver,
Yes,
right?
yes.
Okay. Mm. Okay.
On on the basis of ongoing analysis,
Uh,
we can make a possible conclusion that the exosomes at 8 hours they trigger a strong pro-inflammatory response,
right. We
while
already know about that.
while at 48 hours
Mm.
they are mostly involved in translation process at cytoplasm. Maybe it is related to recovery or the healing response of the liver, which is
Not yet. Not yet. Until now, we know only in hepatocyte, right?
yes in
Only
hepatocyte.
hepatocyte translational, the machinery increase, right?
Yeah,
Only, okay. Mm.
I will perform the non. I'm working
Okay,
on the non-parenchymal.
okay. Mm.
Any questions?
I have a question. So,
Yes,
Imran, why are you doing this experiment? Very simple question. What's this the goal of this experiment?
we want to know the effect of the exosomes on the liver tissue, because once we systematically administer the exosomes, they are mostly concentrated in the liver. So if we are using exosomes as a drug carrier or any other medium of therapy, we want to know what exosomes do to that to the body before we add anything in the exosomes and then perform the.
Are you sure?
Mm, yeah.
Why you're doing this experiment? I mean, I know that it may have like good effect of using exosome, but why you're using exosome for the recovering from the liver? Why? Why you're targeting liver?
Because after the systemic administration, the exosomes are highly localized in the liver than any other organ.
So that means for example, if you have a cancer, liver cancer, then you think exosome treatment going to be the useful or no?
Uh, that depends if we are using exosomes in what capacity. If we are using exosomes as a drug carrier, or we are using any anti-cancer drug tagged with the exosomes, they can give
So
us beneficial response.
I'm asking. So I to go back to the what I asked. If I ask why you're targeting liver for exosome, and second question, the it's going to be good benefit for the liver to using exosome. But what about other organ? It's going to be same effect or no? If you have a cancer with other organ, like if you have like a skin cancer or breast cancer,
We
you
can
say
use we can use exosomes, but we can employ different route of administration, like local administration or the tissue specific administration of the exosomes, then we can expect the same kind of effect.
Hmm. So this result applies to whole other organ or only in the liver?
Since this kind of research is still in early phase, so we cannot make a definite conclusion, but what we are doing, this can definitely tell us like where we are lacking in using exosomes as a drug carrier or exosomes as a means of communication between tissues. Like there are many studies which are working on the cell to cell communication in tumor microenvironment, mostly using exosomes. So we can know how exosomes how much exosomes are involved in this process.
And I have a question for the
Yeah.
UMAP analysis.
Yeah.
Do you think that's the right data or no?
To my knowledge, that is right. If you have any input, I will appreciate it.
So what's the purpose of using UMAP plot?
From the UMAP plot, we can find the cell-specific clusters in our data set, and then we can find the cells which are available in our sample, and we can perform the downstream analysis using that one.
I don't think that's the right answer. Why you're using UMAP plot? UMAP plot, why? Very simple reason.
To show the cell type annotation in our sample, like
No,
what kind of cells are in present in our sample.
no, that's the
No.
answer. The reason you're using that is because when you just do the like sequencing and see the whole gene, and
Yes.
when you see that, that's a flat. But while you're using UMAP, you're looking for three-dimensional effect and the whole liver cell. Liver is cell organ. So if you rotated whole liver and then you can locate it whole gene where your liver like where the gene is located in each place in the liver. That's the reason you're using UMAP.
Okay. Thank
And
you.
also when you're using UMAP plot and then did you sort the data from the raw data to apply UMAP?
Like I made UMAP from the raw data or the filtered data. Do you want that?
Well, normally when you're using the UMAP, there's some data, like data is not some data is not appropriate data, which is like some of them when you do the gene expression data, some of them like something is not right data, and you have to do the pre-treatment, make a preset for the deleting wrong data from the your raw data. Did you do that or no?
Yes. Yeah, I that's what I explained in my second slide where I did the pre-processing and I removed the outliers and the doublets from my data, and then I collected the remaining data in a filtered data set, and then I used the filtered data set for every downstream expression
So
analysis.
did you sort it by yourself or using other program when you do that?
Using 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 Seurat,
슈랏.
yeah, in our studio, I use their standard uh, prerequisites for the filtering out the data set.
Okay, the reason I'm asking this question because the I'm doing a similar thing, but when I using UMAP, I don't use the whole data at the once like for time. So before I'm doing that, I using the small number of data and then I sort it by myself by my hand and then using UMAP whether does that the program is right to process the right data to the apply UMAP. So did you do that process or no?
How do you sort by hand? I didn't understand.
So I choose just a small number of data from the
Yes.
gene, right?
Yeah.
Like location data. I like 10 or 20, not many, like not like 2,000 or 3,000. I just using two like 10 or 20 and then do adding the plot for UMAP for just testing. whether this modeling is right or wrong. Did you do that process before you applying UMAP?
Actually, I don't understand your question, but
임난,
what I did
임난,
to,
임난.
yes,
From the single cell,
yes,
you have at least you said you have a data about at least 3,000, right? 3,000 genes, right?
I have
3,000. On one
the
cell, you have the expression level of 3,000 genes, right? Three,
yes, one
at
one
least.
cell must have at least 2 to 3,000
Right,
genes.
right.
Yeah,
Right.
So you cannot make the spot in UMAP by comparing 3,000 with the other cells 3,000 genes.
yeah,
And Professor Kim Wee's mention is that that much of a gene set cannot gene number cannot be operating UMAP the analysis.
yes.
He always reduced the number of the variables. I don't know. I cannot. I don't know. It is exactly term, but
Mhm.
anyway, variable should be reduced.
Yes,
So the in AI the field, I don't know why, but they call this dimension,
dimension reduction. Yeah.
right? So you right, exactly right. You tried the dimension reduction process for
Yes,
UMAP analysis.
yes.
Then what kind of software did you use for reducing the dimension? Maybe
Actually,
PCI by PCA is that kind of analysis.
yes, I use PCA.
PCA, right?
Yes, yes.
So what program did you use for PCA analysis?
It is also part of this Seurat analysis.
Surat?
Yeah, it has everything from the raw data pre-processing to
Okay.
the differential gene expression and everything.
Hmm.
And to find out if my UMAP distribution is correct or not, I
Hmm.
took the universal markers of each cell type
Yes,
which were
you
available on
showed
the
us.
internet.
Right,
So
right.
from that I can see that the expression of,
Cell marker maybe.
yeah.
Cell
So
marker.
that means that my UMAP identification is correct because it is
Hmm.
100% in relation to the data
Hmm.
available online.
this one.
Yeah,
Okay.
yeah, this way.
음, 그, 잘 모르는데 자동적으로 이게 그 데이터 양이 싱글셀 씩으로 나오는 데이터 양이 그, 뭐, 이게 보통 많은 게 아닙니다, 선생님.
아,
그지?
그래서 제가 얘기한 게 뭐냐면
그,
보통 하기
네.
전에 이거
음.
돌리기 전에 보통 한 다섯 개나 10개 진지 레퍼런스 진 그 있지 않습니까? 그 영향을 받지 않는 진들 있지 않습니까?
그렇죠, 그렇죠, 그렇죠.
그거를 먼저 테스트를 해보고 이게 데이터 로케이션이 제대로 나오는지 보고 그다음에 전 돌리는 이렇게 원래 하거든요.
그게, 이게 자동적으로 아마 데이터 플로우 차트가 있거든요. 그 컴퓨터에서 자동적으로 해줄 겁니다, 이게.
그러니까
넣으면.
그건 아는데 가끔 틀리는 경우가 있어가지고 저 한번 검증을 하고 합니다. 저는 보통.
그래.
어,
근데 저도 이 로우 데이터 갖고 이거 제가 안 해봤거든요. 바빠가지고 임난이 한 건데
어,
이 싱글셀 클러스터링, 유맵으로
어.
클러스터링 한 거 보면 이쁘게 나왔어요.
아, 이쁘게 나왔는데 이게
예,
3D 현실을 돌려서 나와야 되는데 지금
이게 좀 이쁘게 나온 것 같다는 생각이 지금까지는 들고 하나 더 하고 싶은 거는 임난
Yes,
you can make the UMAP for each group of cells.
yes,
I mean I mean uh, for the control of 8 hours, you can also make UMAP the cluster,
yes,
right?
yes, I can.
So you can you can make 4 different UMAP analysis, right?
Yes, now I understand what you are saying. Yeah,
Uh,
I can
then
make.
maybe I don't think so. I don't think the patterns of each the experimental group is not different in UMAP analysis, but who know? Okay, so it
Actually,
doesn't matter.
it
Let's
is same. It is same for each sample.
just show me the
I will.
make.
Yeah, I will
Uh,
show you after them. I
uh,
have really.
okay. I, I want to see that
Yes,
patterns.
yes.
If the
Sorry.
exactly same, no problem.
It's
but
exactly same.
exactly? 오케이.
Yeah.
음.
그 제가 이게 왜 3 유맵 가지고 얘기를 드리냐면 이게 각 그 집마다 그거 해가지고 뎁스 변호를 해서 이렇게 이제 강조하면서 그 나타낼 수가 있어가지고 3디멘션에서 그 농도 그 나온 그 익스프레션 그 레벨별로 이렇게 그 색깔 조금씩 바꾸면서 이게 그 뎁스를 표현을 할 수가 있거든요. 그래서 이게 그렇게 하면 안 될까라는 생각을 해서 지금 얘기를 드린 거였거든요.
가르쳐 주세요.
알겠습니다.
저희도
이거
잘 몰라요.
네이처 바이오 테크놀로지에 나온 건데
음.
그,
오케이.
그런 방법이 있습니다. 하면은 이거 보면 각 진마다 이게 그 어떻게 익스프레션 됐는지 그좀 그레디언트로 보여줄 수가 있어가지고 그래서 아마 재밌을 겁니다. 아마.
아, 진 익스프레션이요?
그 그레디언트로 보일 수 있습니다.
예.
유맵으로 사용하면.
저도 그거 본 것 같은데요.
예,
그 이상한, 막,
아마, 예,
이상한 그래프 본 것 같아요. 근데
맞습니다.
그것도 일단, 그거는 몇 가지, 진 넘버를 몇 가지를 지정을 해야 되는데, 아직 분석이 하나도 안 끝났어요.
그렇습니까?
예. 분석이 다 끝난 다음에, 진짜
아,
중요한 거 패턴을 가지고,
아,
그
알겠습니다.
예. 아직 하나도 안 끝났어요.
요즘
아직
제가 이걸
초반이에요.
많이 하고 있어가지고 사실은 이쪽을 많이 하고 있어서.
전 지금 처음 한 거예요. 처음 한 거.
아,
예.
딴 거 분석이긴 한데 이쪽으로 하고 있습니다. 저희도 사실은.
오케이. 컴랜드 사이트. 오케이. 학습, 질문 있으세요?
어, 없습니다. 조금 더 데이터를 봐야지 저는 나머지
예.
데이터를 봐야지 지금 어떻게 할지 좀 볼 수 있을 것 같습니다. 저는.
Professor Kim,
Yes.
can
음.
you share the pipeline you use for the analysis? I will be really grateful if you can share with me.
Okay, because we're actually modifying the code, but we're
Ah,
using different data. So if you give you data, I can give you some code. But can you use a Python or no?
yes, I can use Python, but so you are using Python and you're using SCMP, right?
Yeah.
Yeah,
So
I
if
did
you
not use SCMP because I prefer using Serath because it is most easy
슈라,
and very well
Mhm.
used.
예,
That's why.
슈라를 제일 많이 써요. 슈라.
Yeah.
So if you have a which protocol you're using with Python, and if you have a code, you just send the email, then our lab happily to modify the whole code and then ask you to apply.
음.
Actually, I have my code, but I want to compare with yours. If you
Okay.
can share so that I know if I'm doing correct or not.
Because I'm not using this for UMap for the gene. I'm using different purpose. So it should be code should be a little bit different.
음.
Okay.
Mhm.
음. 오케이.
Uh,
인난 you today you got you studied you did the live confocal.
Yes.
You finished live confocal study?
Yes, for today. Yes.
You already finished.
Yes.
Okay, you got all the images correctly.
Yeah, I got all the images.
Okay. So did you copy the whole the
Yes. Yes, I copy.
so please rename the
Yeah.
each images, right?
Yeah.
Without the naming,
I
never
will
under.
rename. Yeah.
Okay. Okay. Good job. Okay.
Thank you.
음.
다음 주에는 제가 하겠습니다. 아마 제가 조금 아마 2주 후에 제가 쓸 저기 뭐였지? 과제 관련해서 나오는 내용
음.
좀
거기 과제가 뭐에 관한 거예요?
환경 호르몬 관련해 가지고 그 농도에 따라서 PM 농도에 따라서 병이 어떻게 일어날 거냐 거기에 대해서 예측 시스템 만드는 겁니다.
이거 어렵네. 음.
제가 그 데이터를 가지고 있기 때문에 그거 하고 그거를 좀 하는 친구가 제 동기가 있죠. 동기 친구가 있어가지고 그 친구 도움을 좀 받기는 할 텐데 하여간
음.
그런 데이터가 있는 거 가지고 이제 다음 주에 그 당뇨하고 암 기준으로 아마 설명을 할 겁니다.
네, 알겠습니다. 예.
네네네. 아마
예.
재미있을지 없을지 모르겠지만 하여간 그 임란 지금 하는 거하고 좀 비슷하긴 한데
그렇구나.
그거 그 프로틴 농도를 딱 저기 그 해서 하는 건데 좀 다르게 적용을 시키긴 한데 일단 그거 해가지고 아직 메타데이터를 다 못 뽑아가지고 그걸 그 농도별로 이제 해야 되는데 사실은 제일 좋은 게 중국하고 인도 데이터가 있어야 되는데 중국하고 인도 데이터가 지금 없어가지고 받아야 됩니다
음,
지금.
음.
알겠습니다. 그럼
예,
수고하셨습니다.
예.
Imran did great job.
Thank you. Thank you.
오케이.
근데 샘플 프랩은 송라정 학생이 다 한 거예요.
Yes.
샘플은 송라정 학생이 다 한 거예요. 고생 많이
알겠습니다.
했어요. 이거 하느라고 밤새고 막 했어요. 밤새고.
원래 밤은 새는
이게
거죠.
쉽지가 않아요.
아우,
리버
쉬워 보이지
피팅.
않습니다. 절대
예,
쉬워
이거
보이지
진짜
않습니다.
어려웠어요.
그,
짜증났어요, 진짜. 이거.
그 쉬워 보이지 않습니다, 절대.
엄청 어려웠어요. 송라정 학생이 고생 많이 했어요.
저는 뭐 로우 데이터 받아 딸깍 받는 입장이기 때문에 이쪽은.
만드는 셀 분리하고 그러는데 힘들었어요.
아니, 저도 로우 데이터 만들어 봤기 때문에 알긴 하는데, 할 일이 못 됩니다. 임상 데이터 만드는 게 할 일이 못 돼서 그거 해가지고 아침부터 나가지고 그거 해주고 그거 한 달 해보면 저 애들이 죽습니다. 그럼.
음.
알겠습니다. 그럼 다음 주에 똑같은 시간으로 하고
네,
아마 그 저기 그 뭐였죠? 올해부터 지금 2월까지는 이 시간을 하고 3월부터 시간이 어떻게 될지 모르겠는데
네.
3월부터는 시간
저도 잘
정
모르겠습니다.
네.
스케줄이 어떻게 될지.
그리고 그 저기 이제 그 하는 거 거기에다 이제 드라이브로 발표 자료 다 링크를 해놓을 거거든요. 발표 자료 논문 다 링크를 해놓을 테니까 오픈을 해놨으니까 그냥 보셔도 될 걸로.
네, 예,
알겠습니다.
알겠습니다. 예,
네, 그럼 다음 주에 뵙겠습니다. 그러면.
예,
네, 수고하셨습니다.
감사합니다. 예.
네.